the training data, we obtained the model, we obtained test prediction using the land model and the test data, and we investigated how often molecules were selected, focusing on molecules that were selected at least 25 times. And here I'm showing some prediction accuracy, some prediction estimate, comparing our method to some other methods in the literature. We compare with CIDR, which was also developed by our group, and CIDR is a linear approach. And we also compare it to deep generalized CCA, which was proposed not by us. And we also compare it to regular multi-layer perceptron. So deep generalized CCA is a method for associating different data sets, but then it's a two-step approach, meaning that when it's associating the data, there is no consideration for any outcome, which is different from our approach here. And the multi-layer perceptron, we basically stacked the data set and applied deep neural network to it. So if we look at the accuracy, specifically balanced accuracy, we do find that our proposed method does well. And the Matthew correlation coefficient is competitive compared to some are similar to CIDR, and compared to the deep learning methods, deep GCCA and MLP, our proposed approach seems to perform well. We also did enrichment analysis where we put as input the molecules, and specifically the proteins that were identified more than 25 times in IPA. And we did find that pathways that were related to immune function and pathways that were not related to immune function were enriched in our data set. And we thought that was interesting, specifically the pathways that are not related to immune function. The LXR, RXR activation pathway plays a key role in regulation of lipid metabolism. And our results sort of corroborated what was done in the previous paper, the main paper that generated the data, which found that COVID-19 might be as a result of dysregulation of the lipid metabolism. One other thing that we also found was that the molecular signatures in our lipid candidate list seem to be enriched or associated with multiple diseases, which includes neurological conditions, cancer, and metabolic diseases. And a lot of work nowadays has shown that COVID-19 affects multiple organs. So these findings highlight the need for understanding or learning more about the post-sequelae effect of COVID-19 to avoid, to help understand and address long COVID. So I am done with the one-size-fits-all method, the randomly learned method that we proposed. Now I'm going to shift a little bit of our attention to the case for Bespoke method, specifically method that address the heterogeneity in the data. So again, we are looking at approaches that considers the heterogeneity in different subtypes or different subgroups so that we can identify subgroup specific or subtype specific molecules and also understand or highlight the common molecules. So let's take COPD, which stands for chronic obstructive pulmonary disease. It's a type of lung diseases that affect the lungs of individuals. COPD is known to be multifactorial, meaning that tobacco smoking, genetics, the environment, and other factors can increase one's likelihood of developing COPD. Initially, COPD was thought to be more of smoking, but then it was found that even if some people did not smoke, they were still getting COPD. And so the COPD genetic study was initiated to understand the genetic component of COPD. That was a long time ago, and now there is the emphasis of other omics to understand how proteomics, metabolomics, methylation, and other molecular data work with the genetic data to help understand what's or address COPD. It is known in the literature that males and females have different likelihood for getting COPD. For instance, compared to males, females tend to have faster FEV decline. So FEV1 stands for forced expiratory volume in 1 second. So meaning that how if you were to blow into a spirometer, how far you can blow in 1 second. So it was found that females tend to have faster FEV1 decline, even if they were smoking less compared to males. They tend to have higher airway wall thickness, and they tend to have increased risk for hospitalizations and death. So clearly there is heterogeneity in chronic obstructive pulmonary disease when you are comparing males to females. And so we used that COPD genetic study. We had proteomics and gene expression data. We wanted to understand molecular signatures that increases, that is associated with exacerbation frequency. And exacerbation frequency is another type of outcome for COPD, and which measures it could be when you're coughing more, wheezing, and other symptoms that increases one's likelihood of getting COPD. So our research question here is, we want to develop a method which we are calling HIP to elucidate molecular signatures and mechanisms of exacerbation frequency that is common and specific to males and females. So again, our notation, we are representing the data types by X. Here, we have two data types. We have gene expression, we have proteomics data. Our subgroups here, we have two subgroups representing males and females. Our outcome here, we have exacerbation frequency, which can be modeled using a zero inflated Poisson, because there are a lot of zeros in this particular outcome. But then the method we are proposing can be used for continuous multi-class Poisson outcome. So just like we did before for the round MV learn, we assume again that there is a shared component Z, but here the shared component is subtype specific. So there is ZS for males and females, and we assume that the data can be approximated by the product of this shared component and the loadings. And so we try to estimate this ZS and BDS by minimizing a loss function. And since our goal is to identify common versus subtype specific molecules, we reparameterize this BDS. So this BDS is the factor loadings, and it represents the weight of the variables. And so we reparameterize BDS as element-wise product of the GD times this epsilon D here. If you look carefully at this GD, you will recognize that this GD is independent of the subtype. And this epsilon, this Kisai DS here depends on the subtype. So by forming this GD to be independent of the subtype, we are using the GD to model common effect, so the common molecules. And we are using the Kisai DS to model the subtype specific molecule. And we impose a penalty similar to the L1 penalty, but then we are looking at, because GD and Kisai are matrices, we impose a block L21 penalty. So the block L21 penalty basically first shrinks the rows of the G to form a vector, and then imposes an L1 penalty for sparsity. And so our hierarchical penalty is decomposed as the sum of a penalty that models the commonalities between the different data types, and a penalty that models the subgroup differences. And we use gamma D to allow one to model or to force clinical covariates. So if you have some covariates like age, sex, that you do not want to select, but you want to force those in the model to guide the selection of the molecules, then one can use the gamma D here to enforce that. And we solve an objective that again relates the shared component ZS with the outcome, and we minimize an objective that is a sum of the outcome plus the sum of the integration plus the sum of the loss. And so we are achieving a one-step integration here, and we solve this objective iteratively until convergence using phi star and E star, and we code this in PyTorch. So we applied our method HIP to understand or obtain molecules that are common and specific to males and females, we first did unsupervised filtering to focus on molecules that have potential discrimination. And we divided our data again into training and test split. We apply supervised filtering within the train test, and we built our model on the train and then predicted on the test, and we obtained the genes and proteins common and unique to males and females from the training model. Here I'm showing the prediction based on fraction of deviance explained, comparing our method to other methods. And we do find that our method is quite competitive compared to approaches that basically stacks the data or approaches that ignore the subgroup heterogeneity. This approach here, called self-CCA, was also developed by our group, and it's doing better than other methods. And our current approach, HIP, is quite competitive. So in this, looking at this figure here, we do find that methods that actually model the type of outcome here are doing better than methods that ignore the type of outcome. So our outcome here had a lot of zeros. So if you model it with regular Poisson, or if you model ignoring the Poisson, the zero inflated nature, it might lead to not good result. And so after looking at the molecules that were selected, we did find some commonalities. So here, blue font indicates genes and pathways that are shared between males and females, and red font indicates proteins and pathways that are shared between males and females. We did find that there are common pathways. For instance, the STAR3 pathway, which is common for males and females. The clear signaling pathway was selected just for males, and there were some other molecules that were selected just for males and others just for females. We did a little bit of literature search to position our findings in what is already known, and we did find that some of these molecules have been implicated in COPD research. So one question that you may ask is, what do we do with that? One, it gives us a better understanding of molecules that are common and molecules that are unique to different subgroups. And one could delve deeper into looking at the potential causal implication of these particular methods, and we are exploring that further. So in conclusion, for bespoke methods, I do think that if we are able to integrate all of these different data types, let's say molecular, phenotypic, social determinants of health, and we consider the subgroup heterogeneity, I do think it can provide us with a deeper perspective of the intersections between these different data types. And I also think that these methods can offer greater insight into health disparities. So it will help us understand the differences we are observing in subgroups, and potentially the differential outcomes. And once we know that, it could enable targeted policy interventions to better address these disparities. And from a statistical perspective, these methods increase power for because we are jointly modeling the subgroups as against modeling them separately. Because in the GDs, we are pooling information across different subtypes. So we are increasing power here. I want to emphasize or say a little bit about our app that we have developed that we are calling Multiview Portal. So this is a web app that is currently hosted on Shiny Apps. And the goal of this particular portal is to close the gap between those with limited programming expertise and those interested in doing data integration. And so we currently have linear methods that are ported on this particular app here. But our goal is that any method that we develop, we want to eventually put it here for disease subtyping, for disease prediction. And the rand MVLEN method that I just talked about, we have an R package for that, but it's currently not ported on this particular app. And our vision is for this particular app to be a one-stop place for data integration. And if you were to use this particular app, you can get a lot of figures to understand the complexity or the mechanism in your data set. One figure that I want to highlight is what we call the relevance network. So this particular network gives us the pairwise relationship between molecules and between pairs of different data sets. This is different from looking at just correlation, pairs of correlation, because the input to this particular graph here comes from the multivariate data integration. So we are looking at how variables are connected in relation to in the presence of other molecules. And we could also obtain variable importance if you were to use some of the methods on this particular portal. So we are hoping that this particular portal will be a one-stop place for data integration to advance disease prediction, disease subtyping, offering greater insight into complex disease mechanisms, and potentially identifying targets for therapeutic intention. So concluding remarks, I do believe that data integration can offer deeper insight into complex disease mechanisms. We are generating more and more data types. And so integrating these different data types rigorously can help understand complex disease mechanisms. It is unfortunate that most of the data integration methods and application have only focused on the molecular data. But then the lived experiences of individuals connect or interact with the molecular. And so integrating both the, let's say, the lived experiences, specifically social determinants of health, the environmental data, the molecular data will offer a holistic perspective to complex disease mechanism and will give us deeper insight and help us understand health disparities and better address or improve health equity. And I think that this becomes a team science effort where individuals with different expertise, computer science, computer biologists, statisticians, biostatisticians, social scientists, epidemiologists, all working together to help understand and address health disparity. So I will end here and take questions. I want to say thank you to students that helped with this particular work and my colleague, Dr. Lynn Eberly here. And I also want to say thanks to funding agencies. Our work, most of the methods we have developed, including other deep learning methods, can be obtained on our GitHub page. And if you have questions or interested in data integration, you could always send me an email. Thanks for your attention. I'll take any questions.
Well, thank you, Dr. Safo. Wonderful talk. I'll open the floor for questions.
I do have one from our online audience. So regarding the bespoke analysis, does this sort of approach require knowing the source of heterogeneity? What if there are other unmeasured factors?
Sure. That's a very good point. Yes. So this particular approach requires you to know the subgroup ahead of time. And if you do not know that, one approach that I probably will recommend might be using clustering to first of all identify subgroups and then use this particular method to focus on molecules that are common and also specific to the subgroups. But yes, it does require you to know the subgroups or subtypes ahead of time. Thanks for asking.
I guess I'll pose one question. In your COVID-19 analysis, one of the competing methods was multi-layer perceptron, and it seemed to do okay. One of the issues that comes into my mind when I see these types of models introduced is the notion of the dimension of the parameter space. A multi-layer perceptron is a family of models that can have many different structures and numbers of weights. So do you have a way of comparing model complexity among the different approaches that you took in that case?
Okay, so one, model complexity. I think one thing that you can think about, let's say sensitivity analyses, where you first look at maybe 2, 3, 4 different types of molecules or different types of data sets, and then look at how well the method, all of these methods does or perform, and to hone in on whether adding more data types help improve prediction. It has been done by other people, and they have shown that, yes, indeed, adding different types of data, modeling multiple types of data will help improve data prediction. And so just to understand or address this a little bit better, I think sensitivity analysis might be one way of doing that.
How about runtimes for
Yeah,
that
okay.
analysis?
Yeah, so run MV-learn is actually very fast. And I think especially the individual prediction, and the reason why we use the probability simplex is that we wanted to avoid the need of tuning parameter. And so by that, it runs very fast, seconds, and was even minutes, and you've got it. But then some of the methods, for instance, deep generalized CCA can be very slow, and MLP tends to be fast. CIDR, which was also developed by us, tend to be fast if you have multiple cores. So I would say that in terms of computational time, random VLAN is very fast when you are not doing the group version of random VLAN.
It's 8:45. Any more questions, comments? If not, I'm going to close the session. Thank you once again.
Thank you
Dr.
so
Safo.
much.